All right, welcome to AP Bio. I thought my summer assignment stuff need a little bit of a refresh. I have videos from when I was 27 years old that I have on here, so I decided to make new ones this year. Add some stuff that I think is pertinent and important for you to understand. Remove a couple things that I think that were not as important to focus on, to kind of waste your time on. None of it's a wasting your time. But I did want to create kind of something new and fresh for the way the course currently is, because there have been a few changes. This lecture is going to talk about the nature of science, and then we're going to get into experimental design, biostatistics, how we can use those statistics to determine whether or not there's effects of variables and other variables when we're doing experiments and things like that. And I'm going to try to make this as exciting as I possibly can. I think the stuff's really cool and exciting, but these are skills that you need to know how to do in the AP Bio exam. And I really like to do them over the summer because, we can kind of get rolling right away. There's nothing you have to really memorize. We will practice it a little bit before we have our first test, because this stuff is fair game on the first test. But it's kind of a good summer assignment-y thing to do, because I don't feel like doing this, the first thing that when we get back in September. I want to kind of start right in with the biology stuff. So, what is the nature of science? Remember, so you've taken several science classes to this point. Some of you 3, 4, 5, depending on middle school and things like that. But science is a unique way of thinking, and it came about during the Renaissance, actually, right? During the scientific revolution, and specifically, like, that type of thinking never existed before, which is funny to think about. We didn't design controlled experiments. Philosophy and thinking logically, right, was how we kind of drew conclusions about the world and the natural world back then. But that was ripe with bias, right? So what the scientific revolution did is it came up with a mechanism to explain nature through controlled experimentation in an effort to remove as much bias as we possibly can. Yes, did we still use logic? Yes. But the experimentation and removing bias part was very important. Okay? All conclusions that we draw in science have to come from previous findings, from the data that we collected previously, the conclusions that were drawn previously, and then the new data that we collect, we can then add to what we already know. Sometimes we find out things that were not correct in the past. We have to amend science. Science is a self-correcting process. Over time, if there are mistakes or there are hypotheses or theories that need tweaking, we will tweak them and change them. And that's what makes that this process of science, so powerful. So you may have heard this, the scientific method. You probably learned this in almost every single class, right? We'll go through the different parts of the scientific method, but the goal of science in general is to investigate and understand nature. We want to understand the natural world. I will talk a little bit later about supernatural things and why they're outside of the scope of science, because if they were within the scope of science, they would be considered the natural world. So our job is to try to explain these events in nature and to use those explanations to make useful hypotheses that we can then design experiments to test. We will make observations using our 5 senses as well as technology now, right? We have a lot of technology that we use to collect data, and we will analyze that data. And we're going to talk about the different ways we think about science and eventually get to the data analysis part towards the end of this video series. So the general scientific method, you state your problem. You want to know how something works, right? You research the problem, you figure out that there's this place where we have a gap in our understanding, so we form a hypothesis about that, which is basically, they say the educated guess, but it's like saying, we think that there's going to be an effect of one variable on another, which we'll get into soon. You're then going to, based on that hypothesis, design and conduct a controlled experiment. You're going to collect data from that experiment. You're going to analyze that data, make charts, graphs, tables, and discuss that data and draw your conclusion. And then, if needed, retest, or some other scientist will retest your work to see if they get consistency and reliability in the data that you collected and the conclusions that you drew. And this is just showing you another diagram kind of showing the same thing. You ask a question, you do research, you construct a hypothesis, you test. Is the procedure that you're doing, this is the part that didn't talk about, was the procedure working, yes or no? If not, do we need to tweak it? I know from personal experience, I did have to tweak mine, the experiments that I was doing in college several times to make sure that they worked properly, because I was finding errors and issues with the procedure that I was doing. If the procedure is working, you collect lots of data, you analyze that data. Do your results align with your hypothesis? Do they not? If they do not align with your hypothesis, you may have to reframe how you're thinking about the problem and it's not start over, but maybe tweak the experiment a little bit. Your goal is not to support your hypothesis, right? Your goal is to figure out if your hypothesis is supported or not. You shouldn't be trying to reach a goal of, oh yeah, my hypothesis is correct, right? And then you will communicate the results to this overall scientific community, which is when you publish papers and things like that. That'll go through a process called peer review, which we're going to get into soon as well. But first, we have to talk about hypotheses. This is one of the things you're going to have to be able to do on the AP Biology exam, is to come up with a hypothesis based on information that's given to you. And a hypothesis is a statement based on observed characteristics of an object, but it is not a prediction. Now, the AP Bio exam wants you to include a prediction with your hypothesis. I'm going to differentiate between the two. Remember, on the real thing, and when I assess you, I'm not going to make you know the difference between the two. When I ask you to come up with a hypothesis for an experiment, you will simply give me what's called a full scientific idea, and I'll go over what that entails in a second. So in the past, you've learned this. If, then because. If I do this, then this will happen because an explanation. This one includes a prediction, which is a directionality to the outcome of the experiment, right? Like there's an effect of fertilizer on the growth of a plant. That's a hypothesis. But if I said fertilizer will increase plant growth because nutrients are needed to make certain molecules, things like that, then that would be more of a full scientific idea that includes a prediction. A scientific hypothesis has two qualities. It must be testable. You have to be able to test it using controlled experimentation. And it also must be falsifiable, which means that if we collect data, there is data that we could collect that would falsify the hypothesis or not support it. So you cannot have a hypothesis that's almost guaranteed to be supported, essentially. We could collect data that does not support it, and then we would have to draw that conclusion. So here's showing you kind of the differences between hypotheses and predictions. And at the bottom, I give you an example of a hypothesis and a prediction, and how they're a little bit different. Eating greasy, high-fat foods can cause acne. If I eat healthier foods, then my skin will produce less oil, which then can lead to acne, right? But the way they do hypotheses and predictions and the differences are really just semantics. I know I talk about them in here, but just be aware that there's some kind of, like, even in science, there's debates over when one's more appropriate than the other. So don't worry too much about differentiating between the two, and I'll get you into the kind of how I want you to frame all of your scientific ideas. So the old way, right, to do it was, if I do this, then this will happen, because if I study my vocabulary words, I will do well in the quiz. But we can say the way I prefer to do it is variable A affects variable B. Studying affects achievement on tests, right? Studying will affect achievement on a vocabulary quiz. I didn't predict how it will. Could it be higher or lower? The prediction would then be related to the directionality that I would give to it. So a true hypothesis does not include a prediction, but remember, you always will, when it asks for a hypothesis on the AP exam, include a prediction, because that's what they're looking for. So here's an example. Plants need sources of nitrogen and phosphorus to thrive. It is hypothesized that adding nitrogen and phosphorus-based fertilizer will affect plant growth. When fertilizer is added, one would expect an increase in overall biomass of those plants compared to the control group that received a sham fertilizer or a placebo fertilizer, right? So that is a full-blown scientific idea. It even talks a little bit about our experiment that we're going to do as well. So if you have to do a hypothesis, it's great to include something that includes all of this stuff, because you will always get the point then. So here is showing you the difference between the hypothesis and the prediction. In red is the hypothesis: adding a nitrogen and phosphorus-based fertilizer will affect plant growth. The prediction would be an increase in overall biomass of those plants compared to the control group that received the placebo or sham fertilizer. Okay, so you're going to want to write a full scientific idea when it gives you that prompt to come up with a hypothesis on the AP Biology exam. Okay, so why does science not deal with supernatural claims? So in your notes, you're going to have to listen to what I say and write about it. Remember, and I kind of alluded to this before, if something is supernatural, that means it's outside of nature, which means that we cannot measure it effectively. So supernatural things would be things that would essentially be difficult to show and support with actual evidence. So any supernatural claims, things like religion, things like psychic abilities, things like ghosts, or things that we consider supernatural, are outside of the realm of science. Some people think that, like things like ghosts don't exist, or things, right, like that. And there is some scientific questions there, and people can try to test it. But if people truly think those are supernatural, that means that they are outside of the realm in which we can kind of look at as scientists. We have to presuppose, or we cannot presuppose a conclusion that has no evidence for it. Even for like something when we talk about religion, like the existence of God or deities, we tend to not, in science, we don't deal with that because we have to have evidence for, like, direct evidence of the supernatural being. And if we don't have that, we are not allowed to directly use that information to draw a conclusion. So we always have to kind of start with evidence and build our way up. We cannot presuppose something and go down from it. It's just one of the rules that we are not allowed to do and use. All right, so what is the purpose of a hypothesis, right? It helps to define what we're looking at. It helps us to be focused in our experiment. Like, we should design our experiment this way because this is what we're trying to test. And it helps us to think ahead of what our results will be, and that's really the prediction, right? What are the results that we think will happen, and logically, why do we think that they will happen? Now, this is important. I almost promise you that this will be on your AP exam somewhere. It will definitely be on your tests over and over again. I do this, where it asks for what is the null hypothesis. We will get into this too when we talk about statistics, but basically when there's a null hypothesis, is there is no effect of an independent variable and a dependent variable. So if I said there's an effect of fertilizer on plant growth, the null hypothesis is there is no effect of fertilizer on plant growth. This is not an opposite hypothesis. This is not like saying, oh, fertilizer will affect plant growth, and our hypothesis would be it increases plant growth. The null hypothesis is not that it decreases plant growth. It's literally that there is going to be no difference between the groups. So whenever you think about null hypothesis, you think no effect, right? Fertilizer applied weekly to a plant will have no effect on the growth. It is like our baseline. There will be no difference between our experimental group and what we'll call our control group, which we're going to get into soon. As I said, the null hypothesis is not an opposite hypothesis. There's no effect. If there's slight differences, randomness and probability could explain those differences, not the variable that we are testing. Okay. Now, this is going to sound confusing. The alternative hypothesis, or something called HA, is the hypothesis that you are kind of putting forth, that you think that there is an effect and the directionality of the prediction of that. So that's the alternative. The alternative is not the opposite hypothesis. So let's say you collect your data and you find that fertilizer actually decreased plant growth. That would be something you'd have to kind of retest, and you would say, you would draw that conclusion. It was the completely opposite of what we predicted, and this is some reasons why, and then you can come up with another experiment to test that as well. The next thing that happens after you do your experiment and you draw your conclusions, you write your paper up, and you do a really good job of analyzing your data and all that, you do something called peer review. And peer review is one of the most important parts of science. So once your paper is written, it gets sent out to like 5, 6, sometimes more other experts in your field. And their job is to rip your paper apart. Now, it sounds like it's like, it sounds like it's going to be like a bad thing. This is a good thing. This is why science works. When I was in college, we would have parties. We would have like groups basically in with our professor, where as a grad student he'd get his papers from other scientists, and our job was to rip it apart. So we talk about it and say, like, this is what's wrong with this, this is what we need to fix, this is what we think is, needs to be explained better. I don't know why this conclusion was drawn. You need to explain this further. There's an issue with your experimental design here. All that, and then they send it back, and they either have to fix it or they have to retest some things, they have to redo some things, so that the science is always as sound as possible. So peer review is a really important process for science to be as effective as it actually is. Once it gets accepted, so finally everyone's criticisms and things get taken into account, everything's fixed, your ideas are then used to expand on that area of science. So now you've created an idea, essentially, that no one has ever thought of before. And then new experiments will come from your findings. And if you make a mistake, the people that, in your experiment, for example, or if you're trying to be biased or like fudge your data, people that will then use that information will likely find that error as well. And they will kind of discredit and not support your idea may not have support anymore because we have more evidence to show that that is not the case. So science is a self-correcting process. Some science that is bad does slip through the cracks, but eventually all the important ideas do get fixed because the process works. All right, the next important thing we have to talk about are variables. Again, you're going to have to do this, I guarantee you, on the AP Biology exam and on your first test / your first quiz that you're going to have within the first week and a half or so of school. So your independent variable in an experiment is the thing that I'm testing. So if I'm doing the effect of fertilizer on plant growth, the independent variable is the fertilizer. It is what I'm manipulating as the experimenter to see what the outcome of that is going to be. The dependent variable is the outcome. So, for example, again, the effect of fertilizer on plant growth. Fertilizer is the independent variable. The dependent variable is the growth of the plant. It's what we measure. The way I can remember independent, it's what I change independent. The dependent depends on the independent variable, but it's also the data collected. So if you're looking at a table with data in it, the numbers are going to be your dependent variable. So whatever the numbers are, or whatever the units are that are measuring those numbers, that's going to be your dependent variable. Hypotheses are always written the effect of the independent variable on the dependent variable. So if we look at this data set right here, what are the independent variables and the dependent variables? So I'm going to have you look at that and pause the video. Now again, you don't have a lot of information here, but the other thing that I said, we look at this previously, is that the independent variable is always on the x-axis of the graph, and the dependent variable is almost always on the y. Now, there are some exceptions, but few and far between. Most of the time, this is the case. I have seen the one graph on the AP Biology exam, and it wasn't one you had to make, where those were flipped, and but it was kind of obvious what was affecting what in that experiment. So in this case, we're looking at the probability of nest success in an environment with predators and without. So you can see it's the presence or absence of the cuckoos, right? That is the independent variable. What we want to see is the presence of predators, the cuckoo, which is down here, going to affect the probability of nest success. So the independent variable is the presence or absence of predators. The dependent variable is the probability of nest success, which is the measurement of how many offspring are produced from the nest, I think is what this was, like, or percentage. Okay, so that would be the independent and dependent variables. The independent variable being presence or absence of cuckoos or the predator. The probability of nest success being the dependent variable, and there's an effect of probability on there's an effect of predators on the probability of nest success would be a hypothesis. And the prediction would be specifically that that in this case it would increase, which is weird. That would be the outcome of it, as you can see here, at least based on how we know data so far. We're going to add something to this towards the end of this video series that talks about why this is not enough information to actually draw a conclusion on. The next thing is you have the treatment groups in an experiment. So those are two variables that we test. You have to know those, but then we get to the groups, the experimental and control group. The experimental group is the group that is going to be tested. That is the thing that receives the actual independent variable. There's an effect of fertilizer on plant growth. The experimental group would get the fertilizer. The control group, on the other hand, is what we are comparing things to. There are different types of control groups that I'm going to go over in a second, but the common one is called the negative control, and that is the one that does not get the independent variable. So that would get like the sham fertilizer, because we want to compare that. That's our baseline, okay? So our control group usually gets our placebo, our baseline, or it's our baseline that we're comparing to. It will not get whatever the independent variable is in that treatment. So there are two major types of control groups. There's the negative controls and the positive controls. Usually they ask for negative controls. So negative controls are used to ensure that there is no change no change is observed when a change is not expected. Right, so the negative control kind of is like normal conditions, and then we apply this new condition, which is our experimental group, and we want to see the effect of that new condition. Okay, they do not receive the independent variable. Positive controls are a little different. Positive controls are using experiments to show what a positive result would look like. So if there is like an achievement we're trying to get to, they will ensure that the testing procedure it's possible to get that outcome. Because it's kind of like optimal in that direction. Like it'll show an effect, and this is what the effect should look like if there is a positive outcome. So let me show you an example of this in a way that's kind of simple to understand. So here's an example of an experiment with both. We're just going to test the presence or absence of vitamin C in fruit juice. So our negative control would be water, one that does not have vitamin C in it. And a positive control would be a solution with vitamin C in it, right? That's something that has our positive result. Then we test unknown samples of orange juice to determine is there vitamin C in it or isn't there, and we can compare it to either the positive control that has vitamin C or the negative control that does not. Okay, but neither of which are experimental groups. They're just kind of what we're comparing our experimental group, which would be this third group right here, to. There are also controlled variables. Those are variables that you control in an experiment. Those are different than actual control groups, right? The control group is a group that gets a specific or lack thereof treatment, right, that we're comparing to our experimental group. Controlled variables are variables that we need to keep the same between our experimental and control groups because they could affect the results. So let's think of some examples here. So if you're doing the fertilizer on plant growth, like we should make sure that the amount of water they're both groups, both control and experimental, is the same because we know water affects it. The amount of sunlight could affect it. The temperature could affect it. The other nutrients in the original soil could affect it, right? All of those things could impact plant growth, so we need to keep them exactly the same between the two groups, right? So here's showing you, as I said, I think I just talked about all these things, my bad, right? All of this stuff is important to think about when you are doing a control over your control group and your experimental group. You should only vary in the independent variable. Does that make sense? The only difference between the control group and the experimental group should be whatever your independent variable is. Other things that could affect it need to be controlled and taken care of, and make sure they're the same in both. So how do we improve experiments? One way we can always improve experiments is we can repeat experiments and increase sample size. Those are the two ways. Like if you redo the experiment, we collect more data, it's going to increase the reliability of that data, right? If you flip a coin 2 times, it's possible for it to show up heads both times. Now, is it likely that there's not a 50-50 shot of getting heads or tails? No, probably not, as long as it's a fair coin, but randomness could explain that result. Now, if I do that 1,000,000 times and all 1,000,000 times turn up heads, I would say that there's definitely a skew towards heads somehow on that coin, right? Or, you know, if it's just the more data that you collect, the less chance that statistical noise will dictate the kind of like your outcome, right? So you want to kind of repeat the experiment and collect more data because that'll give us a better idea of the reliability of the sample that we collected. So we always want to reduce bias in our experiments, and bias is an important kind of idea. Bias is essentially, bias is how we as scientists, even if we don't want to, can affect the outcome of the experiment beyond that of the independent variable, right? So, like, even if you're trying to do something as simple as sample insects in a field, right? And if we're collecting, if we're like going to use information, like we collect them from, you know, we're extrapolating to the whole field, but there's a bunch of flowers in one area where those insects are more likely to live, and we just count that area and we don't count the other spots, that's going to kind of give us and then we extrapolate that to the whole area, that's going to give us a biased sample, you know? So you want to try to remove that. And sometimes there's experimental biases, like they're completely unintentional, but they still can creep in. You may have heard of like double-blind studies where both the experimenter and the participants have no idea what treatment the participants are getting versus the like who's getting the control, who's getting the experimental group. No one has any idea. The experimenter doesn't, and the people who are getting them don't. And they won't find out until after all the data is analyzed. So, again, how do you always improve it? Redo the experiment, increase sample size. Also, if there's some variables that can be affecting it, that would be part of the reduce the bias thing. We need to control for those between our control groups and our experimental groups. Next important thing you need to understand, what is a theory? A theory is a hypothesis that has major explanatory power, right? It explains a wide variety, explains a wide variety of phenomenon, and it is shown over and over again that this hypothesis that has explanatory power has never been shown to be incorrect. Not just like, you know, this is a hunch. A theory in science means something completely different than a theory in our general use of the term. That is why when people say things like, oh, the theory of evolution is just a theory. Well, so is cell theory. It's just a theory that everything's made up of cells. And the Big Bang theory is just a theory. And germ theory, which is the idea that bacteria and viruses cause illness, is just a theory. We have never shown those things to be incorrect, ever. And in fact, there's differences between theories and laws. A theory has explanatory power. So there's both a theory of gravitation and there's a law of gravity. The law is directly measurable and it's always the same. The theory is how gravity works. That's where Einstein's theory of special and general relativity comes into play. It explains why gravity happens versus the fact that we know gravity is a thing, which is the law part of it. Okay, so theories are not like we generally use them in terms of the term. It is supported over and over again. It's never shown to be incorrect, and it has a wide variety of explanatory power. It explains things. So it's broad in scope. It helps to generate new hypotheses, right? And as I said, it is supported by a ton of evidence. And if, let's say, something comes up tomorrow and it shows a theory to be in question a little bit, we have to change the theory. We will probably make sure and check for the validity of the experiment that kind of holds it in question, because everything to that point has shown it to be correct. So if you're going to change it, we kind of need a lot of evidence to do so. But science is self-correcting. It will eventually figure that out if our theory is no longer supported and no longer correct. So the scientific method, as we talked about before, is an idealized process of inquiry. Obviously, it's not as simple as like, oh, we do this step-by-step type of thing. You know, it doesn't have to follow exactly those orders of steps, though it usually kind of does in most instances. But the textbook version of it is very oversimplified, I would say. All right, the next thing you have to be able to do on the AP Bio exam. So you have to be able to identify independent dependent variables, come up with hypotheses, design an experiment, both with control and experimental groups, and talk about things you would need to control in an experiment to improve the validity. Another thing they'll ask you to do is to ask a scientific question. A scientific question is simply a question that you will generate based on some prior scientific knowledge. It tends to be more exploratory and broad than a hypothesis is, which is more of a statement. A question mark at the end of it would be a scientific question mark. A statement would be more of a hypothesis or a prediction. Now we can develop hypotheses based on questions, which tend to be more broad. So, for example, what are the effects of plastic waste on marine ecosystems? That's a scientific question. What are they? Let's come up with a hypothesis. Marine ecosystems that experience high levels of plastic pollution, this is a hypothesis with prediction, by the way. Marine ecosystems that experience high levels of plastic pollution exhibit a substantial reduction in biodiversity. So now this is a little bit more focused, right? The hypothesis is more focused. The question is a little bit more broad. What is the effect on marine ecosystems? Our hypothesis is going to look specifically at biodiversity and the impact of plastic pollution on that. So let's see if we can come up with a hypothesis or scientific question based on either the question or the hypothesis. So the first one, we're going to give a scientific question. How does lifestyle affect the risk of heart disease in adults aged 40 through 60?